Danny and I are very pleased to introduce our mass spectrometry capabilities to you, and especially our high throughput systems, the rapid-fire mass spectrometry and the ECO-MS. Let me situate its finding in the drug discovery journey. The goal of the drug discovery process is clear. It's to identify successful preclinical candidates with drug-like properties in the shortest period of time. Successful preclinical candidates are ultimately selected by employing the most robust and reliable hit-finding strategy. The drug discovery journey is highly structured with a well-defined sequence of steps. Target validation, hit-finding, hit-to-lead, lead optimization. All of these phases can be supported by our Eurofins Discovery services. The topic of today's webinar is mass spectrometry, and especially why and how to use mass spectrometry in drug discovery. When we think of mass spectrometry, we think of a label-free technology, so no labels on the substrate or proteins, as is the case with most fluorescent readouts in HTS. The other advantage of mass spectrometry is that makes it possible to monitor several transitions in one injection, such as substrates, product, or internal standard in one go. MS also saves time and money, as there is no need to repeat the experiment with an endogenous substrate after performing HTS using a modified reporter system. MS can support screening, profiling, and mechanism of action study for a long period of time. The limitation of the mass spectrometry has been the throughput. But as you will see in this webinar, this problem has been solved by innovative technologies. As I just mentioned, we can now tailor mass spec with the throughput needed for your project phase. For the early phases of the primary pharmacology, that is the HTS and hit confirmation phase, the ECO-MS and the rapid-fire can be used for a throughput varying between 30 to 50,000 compounds per day for the ECO-MS to 7,000 compounds a day for the rapid-fire. As the project moves forward in the second pharmacology phase, including the MS binding, mechanism of action, and pharmacokinetic phases, both rapid-fire and standalone LC-MS system can be used for a lower throughput. In this webinar, you will hear from both Danny and myself. To give you a brief introduction, I'm Céline Legault and I'm a partnership director. I joined Eurofins Discovery 3 years ago, and I'm extensively involved in the design of screening cascades for hit finding, hit to lead, and lead up. Previously, I was a scientific project leader in the screening department of Institut de Recherche Servier, designing and running screening cascade for HTS, hit to lead, and lead up. Danny?
And my name is Danny Gendifouk. So I'm a mass spectrometry engineer for 2.5 years at Eurofins. I have an expertise in UHPLC, MS/MS, RapidFire-MS, and ECO-MS method development. I also an expertise in enzymatic reaction and MS binding mass spectrometry. Previously, I worked on several R&D projects in the academic sector to develop and optimize a new methodology using UHPLC-MS experiments to identify and quantify isomeric or isobaric compound in mixture.
In this webinar, to start off, we will present to you how we develop experience with the ECO-MS. Then we will present an HTS proof of concept on the ECO-MS, and finally, we will present another mass spectrometry application, MS binding.
So before getting to the heart of this webinar, I would like to say a few words about Eurofins Discovery's different analytical systems. So we have multiple platforms which can be used for sample analysis with low throughput, with the LC-MS/MS systems to analyze between 100 to 500 samples per day. Then we have systems for medium throughput with the rapid-fire system with up to 7,000 samples per day, and a system for high throughput with the ECO-MS with up to 30 to 50,000 samples per day. So in this webinar, we will focus on rapid-fire systems and ECO-MS system. So whole rapid-fire-MS system is ideal for medium throughput and particularly useful for screening of small to large compound sets. This makes it a great tool for it-to-lead and lead optimization programs. It can analyze up to 7,000 samples per day with an injection every 10 to 12 seconds. By combining it with a triple quadruple, we can monitor up to 5 transitions simultaneously, which reduces the risk of false positives. Depending on the sensitivity needed, the RapidFire can also be combined with either triple quadruple mass spectrometer for sensitivity or Q-TOF mass spectrometer for high resolution. We have already screened 90,000 compounds with a 15-day TAT. And this year, we added a second RapidFire mass spectrometry to our labs. Our EcoMS system enables us to perform for high throughput projects as it combines an acoustic droplet ejection system with a triple quadruple mass spectrometer. This system is ideal for enzymatic HTS assays. These main benefits are its speed, reproducibility, and affordability. EcoMS can monitor up to 3 transitions simultaneously in one ejection, perform up to 1 ejection per second, and when combined with a robotic platform, can analyze up to 100 plates per day, so around 30 to 50,000 samples per day. The advantage of EcoMS is to be able to miniaturize assays. The volume of enzymes and buffers needed compared to RapidFire-MS system can be divided by 2, in addition to the fact that by performing the assay directly in the EcoMS plates also allows for a reduction of plastic waste. So in this part, we will focus on your EcoMS capabilities. So the EcoMS system employs a technique called acoustic droplet ejection, or ADE, in which ultrasound waves are used to propel nanoliter scale droplets from a sample into a system called open port interface, or OPI. Within the OPI, the droplets are diluted with a carrier solvent in order to ensure that the background interference from abundant biomolecules in the sample is no longer an issue. The OPI then sends the diluted sample through a transfer capillary into the mass spectrometer with electrospray ionization for analysis. The EcoMS has a speed of execution of up to 1 sample per second. This high throughput system multiplies the analysis speed by 60 when compared to the analysis of a 384 well plate in LC-MS, and by a factor of 6 when compared with the RapidFire-MS system. This system does not require liquid chromatography and greatly reduces sample and solvent consumption due to the ejection of only a few nanoliters. The EcoMS has a high tolerance for complex matrices, even in the presence of detergents and extracted plasma. They have the same detection limit and linearity thanks to the sample dilution using a carrier solvent, and there is no carryover between each ejection. So on this slide, we would like to present how we develop an enzymatic assay on the EcoMS. So first of all, we show the impact of the buffer in order to know if the buffer has some interference with the mass ionization of the product. Then that of the carrier solvent and flow rate optimization to obtain the thinnest and most sensitive peak possible. The nanodroplet optimization to be ejected to increase the SNR, and the limit of detection and quantification of the product. We determine the quantification of detection limit for all products, and we perform kinetic reactions to determine enzymatic parameters with Km and Vm value. We created our own database to optimize the assay development time and to reduce the cost. So to create our database on the impact of the buffer on the EcoMS, we analyze 15 products with 8 different buffers, including detergent, plasma, and salt, with 12 different carrier solvents to have the real impact of the buffer on the mass ionization. In this 3 table, we have included 3 carrier solvents with 3 different additives, so formic acid, ammonium acetate, and ammonium fluoride. In red, you have the compound with either no or weak signal detection, up to the green with strong signal detection. We noticed that some positive results were observed using organic solvent containing water and 0.1% formic acid. For example, with propranolol, which has a strong signal with buffer 3 and 6, whereas a significant amount of signal loss was observed using the same carrier solvent with ammonium acetate and ammonium fluoride as additives. With the results obtained with the carrier solvent containing formic acid, we try to add a detergent to each buffer used in order to have better solubility of the sample in each well. You will notice that after the addition of detergent, a positive impact was observed for several products. For example, with anacardic acid, where no signal was obtained with different buffers, whereas after addition of the detergent, we could observe a clear improvement of the signal for each buffer. We see that the detergent facilitated an increase of the solubilization of the product and reduced the potential interaction of both, so the transfer capillary and the plastic of the plates. So this database enabled us to create a decision-making matrix, facilitating the choice of the correct carrier solvent to use according to the buffer and the chemistry of our clients' products. It also reduces the development time during the optimization of the EcoMS method and thus the development cost. So following the study of the buffer effect, we optimize the carrier solvent for each compound and buffer. The carrier solvent pushes the sample into the transfer capillary to reduce any interaction that the product might have with the transfer capillary, which in turn could induce larger peak, so longer analysis time. We tested several carrier solvents with organic solvents, water, and with different adducts with acid and base in order to optimize the flow rate for each solvent. So here we show you how we conducted the optimization of the carrier solvent flow. So we increase the flow rate of the carrier solvent until a decrease of the signal was observed. So here at 450 microliter per minute, or until the observation of an OPI leak at 500 microliter per minute, stopping the EcoMS systems. Then the flow rate with the best SNR was selected. However, we realized that increasing the flow rate can lead to an increase of the background noise. We therefore optimize the flow rate of several carrier solvents with different additives shown in this table. So with formic acid, ammonium acetate, ammonium fluoride, DDM, TFA, for example. So the next step was the optimization of the number of droplets having a direct impact on the SNR. Indeed, if the buffer interferes with the mass ionization of the product, it is not recommended to increase the number of droplets ejected towards the MS, because a larger volume of buffer will induce an increase of interference with the mass ionization and thus decrease of the SNR. On the contrary, if the buffer does not interact with the mass ionization of the product, an increase of the number of droplets makes it possible to increase the SNR significantly, and at the same time the limit of detection and quantification of the products. So in this table, we show that for certain products in a basic buffer, for example, with antipyrine and colchicine, the limit of detection and quantification is improved by a factor of 2 for colchicine and by 5 for antipyrine, with a droplet ejection of 20 nanoliter than 2.5 nanoliter. So the last step of the EcoMS method development was a calibration curve to determine the limit of detection and quantification of the product. Here we demonstrate that for each molecule, so for example, diltiazem, that the limit of detection and quantification value are very similar, whether in a basic buffer, in presence of detergent, and in an extracted plasma. So this shows that complex matrices can be analyzed in the EcoMS without a pre-cleaning step, thanks to the dilution of the sample in the transfer capillary. So in conclusion, concerning the EcoMS method development, we offer a 5 business TAT to develop the EcoMS method development for your products, including the impact of the assay buffer on the mass ionization of the product and the MS method development. Then the development of the ECO method with the optimization of the carrier solvent, its flow rate, and the number of ejected droplets. And we finish by the determination of the limit of quantification and detection of the product in the assay buffer. So now we will focus on the enzymatic assay development, and particularly with the determination of Km and Vm values with the kinetic reaction performed directly in the EcoMS. So we will now show you to define enzymatic parameters, so Km and Vm values with the EcoMS, including of substrates with different chemical properties and enzyme classes, allowing you to reduce assay setup time and the enzyme consumption by performing live kinetics directly with the EcoMS. So we can do kinetic reactions for well-known enzymatic reaction at Eurofins with the CONT for catechol O-methyltransferase assay with only one well in the plate by ejecting X droplet every Y seconds, enabling us to obtain a significant amount of data, as well as to plot a kinetic curve at a substrate concentration. As the kinetics are done directly in the EcoMS, we reduce the technician time, and this decreases development costs. Therefore, we can carry out kinetics on 12 different concentrations with only 12 wells, facilitating the determination of the true value of V0 by placing in the linearity range with the highest slope. As the results, we can determine the true value of Vm and Km with precision, all with low enzyme consumption for each enzymatic assay. To demonstrate that our live kinetic method on the EcoMS works, we present a second case study performed on a second enzyme for the CYP3A assay in the presence of bactosome in the assay buffer to determine the value of Km and Vm. So here we show in 2 case studies that the EcoMS increases the robustness of the kinetic reaction and is instrumental in saving lab technician time. We are able to perform kinetic with a true determination of Km and Vm values for highly hydrophilic compounds, as well as for compounds in the presence of bactosome. So other family of compounds are in the process of being analyzed to extend our knowledge and ability to perform kinetics on an even wider range of products. So to sum up, regarding the EcoMS, it has many advantages in regard to the speed of analysis and sample dilution. the rapidity of the development of an EcoMS method, poor ability to quickly define the enzymatic parameter of substrate with the live kinetic directly in the EcoMS, and the decrease of assay setup times, all results in cost savings as well as the reduction of false positive linked to MS technology. However, there are still challenges to overcome with sensitivity and buffer compatibility when the buffer interferes with mass ionization or if the product interacts with the transfer capillary. So drawing from this experience, we can now offer the development of enzymatic assays in 15 business days, including 5 days for EcoMS method development and 10 days for enzymatic assay development. So in this case study, we show the challenges of the EcoMS by comparing the assay development of PIM3, which is a phosphorylated peptide on the ADP Glow system versus the EcoMS. At first, we tried to develop the EcoMS method development for the peptide in the assay buffer. Unfortunately, we anywhere not able to detect the peptide with the different carrier solvent and different droplet ejection volume tested. No signal of the phosphorylated peptide was detected due to the non-specified interaction of the peptide with the transfer capillary, in addition to the buffer compatibility which interferes with the mass ionization of the peptide. We thus turn to the RapidFire-MS system to continue the PIM3 assay development. So as the EcoMS development of the phosphorylated peptide was not possible, we chose to use the RapidFire-MS system. So we performed the RapidFire-MS method development for the phosphorylated peptide in the assay buffer containing MOPS, beta-glycerophosphate, MgCl2, EGTA, EDTA, and DTT. So the peptide was well detected using RapidFire-MS system with a limit of detection of 0.5 nanomolar and a limit of quantification of 1 nanomolar. So the RapidFire is able to detect the phosphorylated peptide in the assay buffer because compared to EcoMS system, the RapidFire performs sample cleanup to reduce matrix interference, which in turn allows for greater sensitivity in the mass spectrometry. The RapidFire-MS provides better sensitivity but has lower throughput than the EcoMS. So we perform a screening of 5,000 wells by DRC for this study with a 2 days TAT. So in conclusion, we have shown that if EcoMS does not detect a product due to buffer interference, we can always switch to RapidFire-MS system while keeping a medium throughput.
Thank you very much, Danny. To run an HTS project on the EcoMS, we first needed to automate the EcoMS. So we designed a compact mobile robot with IRS to feed the EcoMS. This platform was designed around a precise flex robotic arm, a room temperature storage of 22 positions, but we could go up to 69 using an extra position in the door. This platform was also equipped with a pillar and sealer, a barcode reader, a centrifuge to make sure the aggregates are stuck at the bottom of the well, and a shaker. Agitation is really important in order to achieve a perfect meniscus, perfect propagation of the acoustic wave, and so a good ejection of the drop. The primary use of the mobile robot is the EcoMS, but we also wanted for it to be compatible with other devices, in particular the flipper penta dedicated to HTS, and also one of our standalone Eco. You can see here the extra position in the door and also the mobile robot with the flipper penta on the right. Unfortunately, as Danny mentioned, the original target we picked up for the proof of concept refused to fly. So we chose to work with another enzyme, the COMT, which stands for catechol O-methyltransferase. COMT inhibitors are already in clinic with applications in Parkinson's disease. However, they show some limitation. For this proof of concept, we ran a small HTS with a subset of compounds from our library of 50,000 of compounds. On the left side of the slide, you can see a quick summary of the assay development and especially the enzymatic kinetics. As Danny mentioned earlier, we ran the kinetics from one single well and we confirmed for up to over 280,000 measures. We generated a considerable amount of data points, which are all illustrated in this regression curve. In the lower part, you have the reference compounds tested on the two systems, the EcoMS and the RapidFire-MS. Data are perfectly aligned between the two MS systems. On the right side, you can see the pilot study data. The assay is robust, as shown with an SNR and shown on the SNR and Z prime monitoring graph, with a Z prime around 0.8. Looking at the distribution of the compounds, we have a Gaussian-like distribution shape, and we didn't identify any BAs. In the end, we obtained a hit rate of 0.17%. We then run the full screen of the 50,000 compounds. Assay is still as robust as the pilot screen with a Z prime of approximately 0.8. The hit rate of the single dose screening is of 0.4%, and from this we selected 200 compounds for dose response curve confirmation. Based on the dose response curve confirmation, we confirm a limited number of compounds with PIC50 distributed from 4.7 to 5.19 of PIC50 value. While working on the proof of concept, we were able to fully explore both the EcoMS and the RapidFire. The EcoMS can be used to accelerate assay development, especially for the kinetic studies. We also showed that the system generates robust data, perfectly adapt to HTS. Having the mobile robot will help us to maintain a satisfying throughput for HTS. As well, we also use the mobile robot during the assay development phase, which also greatly improved the assay development phase and turnaround time. We will now discuss MS binding and its interest for complex membrane proteins such as GPCRs or ion channels. The gold standard for binding assay is radioligand binding. Even if it's a robust and trusted method, it has certain limitations, such as the dependence on the availability of the radioligands. And for novel molecules, they must be radiolabeled, as well as the molecule has to have the appropriate pharmacological properties. In addition, when working with tissue samples, the density of the endogenous target can be a limitation. MS binding is based on the principle of the radioactive binding assay. The first step is to confirm the detection of the compound of interest in mass spectrometry. Compounds and membrane are then incubated. After incubation, the sample is washed to remove the unbound fraction of the compound, and the bound fraction is eluted using an organic solvent. Molecules of interest are then measured by mass spectrometry. Using the method I just described, we measure the direct binding of the molecule of interest by measuring the K-on and the K-off, possible by dilution or competition, and then we are able to calculate the KD. Using a reference compound as a probe, we can also determine the IC50 of any molecules, even if they are not mass spec compliant. Measuring the IC50, we compare it to the IC50 value obtained by radioligand binding. As you can see on the graph on the right part, there is a perfect correlation between the two methods. It makes MS binding a good alternative to the conventional radioactive assay for the characterization of your compound of interest. We bring technology at even higher level of complexity by combining and monitoring up to 10 targets in the same sample, in the same well. And in this case, the 10 targets were expressed in rat cortex tissue. We use 10 different probes to monitor these 10 targets. As you can see on this chromatogram, each reference shows a unique spike. To summarize, we can perform high throughput profiling as it has been implemented on the RapidFire and generate the kind of heat map shown on the right part in order to determine the on-target and the off-target profile of your molecule of interest in one shot. It's an important tool for the target engagement, comparing KO versus wild type and also healthy versus disease animal models. In addition, we can customize the panel in order to establish a relevant off-target panel connected with a specific indication or animal model. So, in conclusion, mass spectrometry is the gold standard of the label-free technology and is a highly versatile technology which is adaptable and to project needs. During this webinar, we showed how we can access a diverse level of throughput using either classical LC-MS, the RapidFire or EcoMS. Each technology has their own advantages and limitations. And thanks to our experts, we can apply MS to screening and profiling of mechanism of action studies, and we can also use MS on other platforms such as ANME or proteomics. Our MS technologies are totally complementary to our other biophysics methods, including SPR, MS-T3 spectral shift. For all of these applications, we have a large park of a spectrometer to cover all of our needs, from high throughput to high resolution mass spectrometers, and for injection of complex samples, as with the UHPLC, to the more simple samples on the EcoMS. Finally, by combining our expertise in mass spec and in drug discovery, we are able to identify the most relevant mass spectrometry system for a specific drug discovery program, and to include these technologies in customized cascades for hit finding, hit to lead, and lead ops program. If you would like to learn more about our global mass spectrometry and biophysics capabilities, or about hit finding and integrated drug discovery services, please visit our website or contact us at hitfinding@eurofins.com. Thank you very much for your attention, and we will stay on to answer any question you might have.